Hello everyone, a very welcome to VYAS Edification Quota. So today in the series of NCERT discussion, we are going to talk about a new chapter from class 12, that is human health and diseases. Human health and diseases. So basically we have total 17 questions in this chapter, and each and every question we are going to deal in this session only. So the very first question of this chapter is on the screen, which is asking what are the various public health measures which you would suggest as safeguard against infectious diseases. Alright, so for that we have to maintain public as well as personal hygiene. Alright, so maintenance of personal as well as public hygiene is very important as a safeguard against various kind of infectious diseases, right? What is most important? Public as well as personal hygiene. Both are very much important and they act as safeguard against various kind of infectious diseases. If we talk about personal hygiene, it includes keeping our body clean. Personal hygiene includes keeping the body clean, also consumption of clean drinking water and food. So this is what we have in the personal hygiene. If we talk about the public hygiene, then it includes proper disposal of waste and excreta, also periodic cleaning and disinfection of water reservoirs such as pools, tanks, etc. Observing proper hygiene in public catering is also very much important and is included in the public hygiene. Now, in case of some diseases which can be spread through air, such as common cold and pneumonia, one should avoid the close contact with the infected person and to their belongings. And in case of diseases which spreads or transmit through insect vectors, the most important measure is to control or eliminate the vectors and should avoid the breeding places of them. So this is what we have to take as measure, and they act as safeguard against various kind of diseases. Now let's write the answer number 1, and we will write it like this. Answer number 1. Let's start with the maintenance. So maintenance of personal and public hygiene. Maintenance of personal as well as public hygiene is important, is very important as safeguard against various infectious diseases. Right? Now, if we talk about personal hygiene, what does it include? So, talking about personal hygiene includes keeping body clean, keeping the body clean, Second is consumption of clean drinking water and food. Right? So this is what we have in personal hygiene. And then if we talk about the next one, that is public hygiene. So public hygiene includes some of the measures like proper disposal proper disposal of waste and excreta, then periodic cleaning and disinfection of water reservoirs such as lakes, pools, etc. Lakes, pools, etc. Also, in some cases of diseases which can spread through air, we should avoid the close contact with the infected person, and to their belongings. Also, we should control or eliminate vectors which are responsible for causing some diseases. So we should control or eliminate vectors along with avoiding their breeding places, right? So these are some measures or safeguards against various kind of infectious diseases. And with this, we have completed complete solution for question number 1. Now moving on to the next question, that is question number 2. In which way has the study of biology helped us to control infectious diseases? Now let's talk about how biology or study of biology actually helped us to control infectious diseases. So biology provides us knowledge about causative agents or causative microorganisms of various kinds of communicable as well as non-communicable diseases. Also, it provides the knowledge or information about mode of transmission of various kinds of diseases, the life cycle of pathogens, and the vectors of pathogens. So biology or the study of biology provides us so much knowledge, so much information about all these things, right? Now with this information or with the help of all this information, this can help us to avoid some causes of diseases, right? And also it helps us to prevent ourselves from various kinds of diseases. Also, even at the time of infection, taking proper food, keeping our surrounding clean, isolation, medications, vaccinations speeds up our recovery. Now, the development of vaccines for many diseases eradicated some or few diseases such as smallpox from the population, right? So this is how biology actually helps us in controlling or avoiding the infectious diseases. Now let's write the answer of this question number 2. Answer number 2, we'll be writing like this: biology provides knowledge about the causative agent or organism of various communicable and non-communicable diseases. Right? So, biology provides us so much knowledge, so much information about causative agents or causative organisms of various kinds of diseases, right? Along with the mode of transmission, along with the knowledge of mode of transmission of various kind of diseases. And also it provides us knowledge about the life cycle of pathogens, and the vectors of pathogens, and the vectors which are responsible for spreading diseases. So it also provides the knowledge about vectors of pathogens. Now, with all this information, we can avoid the tentative cause of diseases. Also, we can prevent ourselves by taking proper care, right? Even at the time of infection, taking proper food, keeping the surrounding clean, isolation, medications, proper medications, vaccinations, speeds up the recovery of a patient, right? Now, the development of vaccine for various diseases eradicated few ones, such as smallpox, right? The development of vaccine of smallpox disease eradicated this disease from population completely. So this is how biology actually helps us in avoiding or preventing infectious diseases. So with this, we have completed the solution for question number 2. Now moving on to the next question, that is question number 3, and is asking, how does the transmission of each of the following diseases takes place? Amoebiasis, malaria, ascariasis, and pneumonia. So we have to give the mode of transmission of each of the diseases mentioned on the screen. First, we are starting with the amoebiasis. Now, how does this disease, amoebiasis, actually transmit from person to person? By ingesting quadrinucleated cyst of Entamoeba histolytica, which is the causative agent or causative organism of this disease, right? By digesting quadrinucleated cyst of Entamoeba histolytica with the food, right? This is how we get this disease. Now, from where this cyst actually comes in the food and water? The cyst of this Entamoeba histolytica comes in food and water from the feces of the patient, right? So this is how we get this disease. Let's talk about the mode of transmission of malaria. Now, if we talk about how malaria or what is actually the causative organism of malaria, that is Plasmodium. Malarial parasite also is the name for Plasmodium. Plasmodium is the causative organism or causative agent for malaria, which is a disease. Now, Plasmodium is transmitted from infected person or from patient to a healthy person when the healthy person is actually bitten by the female Anopheles mosquito. Female Anopheles mosquito is the vector organism for this malarial disease, and the causative organism for malaria is Plasmodium. So this is how we get malaria in a person in population. Now if we talk about the mode of transmission in the third disease, that is Ascariasis. How does it transmit from person to person? This is transmitted, Ascariasis is transmitted from person to person by contaminating food, by actually contaminated food and water, which is containing the contaminated food and water is actually containing embryonated eggs of Ascaris. When we take this kind of food, contaminated food and water, this is how we get Ascariasis disease. Now, let's talk about the last one, the mode of transmission of last disease, that is pneumonia. We all know that pneumonia is actually a bacterial disease, and it can be transmitted by sputum and droplets from the coughing during the coughing of patient, right? So this is how we get these diseases. Now let's write answer of these diseases. This question, sorry. So answer number 3, if we talk about the A1, that is amoebiasis. Amoebiasis. We have to tell about the mode of transmission of each of these diseases. So if we talk about the very first disease, that is amoebiasis, we know that how does it spreads. So by ingesting the quadrinucleated eggs, quadrinucleated, not eggs, quadrinucleated cysts of Entamoeba histolytica. Entamoeba histolytica. Right? So by ingesting the quadrinucleated cyst of Entamoeba histolytica with food and water, Now this cyst spreads in food and water from feces of patient, from feces of patient, right? Now let's talk about the next disease, that is malaria. Now we have to tell about the mode of transmission of malaria. First of all, Plasmodium. Plasmodium is also known as malarial parasite. Malarial parasite is transmitted, this Plasmodium is transmitted from an infected person to a healthy person, when bitten by female Anopheles mosquito, female Anopheles mosquito. Right? Now let's talk about the third one, that is Ascariasis. Ascariasis is a disease which is transmitted through contaminated food and water, contaminated food and water, which is containing embryonated eggs of Ascaris. Right? So this is how we get this disease. The last one we have is pneumonia. Now, first of all, this is a bacterial disease, and if we talk about the mode of transmission, then it transmits with the sputum, and droplets during coughing of patient, right? So this is how the diseases such as amoebiasis, pneumonia, malaria, ascariasis actually transmit from person to person. So with this we have completed the solution for question number 3. Now moving on to the question number 4, and this question is actually asking what measures should you take to prevent waterborne diseases. Alright, so waterborne diseases such as typhoid, chlorella, hepatitis B, etc. are actually transmitted by drinking contaminated water. Waterborne diseases spreads or transmit from person to person by drinking contaminated water. So we have to take some measures, we have to take some care so that we can prevent these waterborne diseases. And for that we have some measures which can control waterborne diseases. What are them? First of all, the drinking water should be clean and free from contamination. And how this can be achieved? So this can be achieved by filtration, boiling of water, sedimentation and chemical treatment of water. The next measure we can take to avoid waterborne diseases is we should not allow the water to stand for a long time, as it can become breeding pool for some microorganisms or pathogens. Then the water reservoirs or water resources should be treated and disinfected properly and periodically. Then the proper maintenance of standard practices of hygiene should be maintained, right? Standard practices of hygiene should be strictly maintained in the public catering. So this is how we can actually prevent waterborne diseases, by spreading in the population. Now let's write this answer, and we'll be writing like this, that waterborne diseases like typhoid, chlorella, hepatitis B, etc. spreads by drinking contaminated water. water. So this is how waterborne diseases actually spreads in the population. But we have some measures. Now, the following measures are required to control the transmission of waterborne diseases. What are these measures? First of all, the drinking water should be clean. The drinking water should be clean and free from contamination. Free from contamination. Right? And this can be achieved by filtering water, by boiling it, by sedimentation and chemical treatment of water, right? So this can be achieved, by filtration, boiling, sedimentation, and chemical treatment of water. And chemical treatment of water. Then the second measure by which we can actually prevent the transmission of waterborne diseases is water reservoirs. Water reservoirs should be disinfected periodically. The third measure is water should not be allowed. Water should not be allowed to stand for a long time. As it can become breeding pool for some of the pathogens. Now, standard practice of proper hygiene should be strictly maintained. In public catering. Right? So these all are measures by which we can prevent waterborne diseases, and with this we have completed the solution of question number 4. The question number 5 is asking, discuss with your teacher what does a suitable gene means in the context of DNA vaccines. So if we talk about what is a suitable gene with the context of DNA vaccines, then a suitable gene refers to the specific DNA segment which can be injected into the cells of the host organism. Now, this specific DNA segment produces a specific type of protein which is helpful in killing the causative organism or causative agent of the specific disease in the host organism's body, and also it provides immunity to the host organism. So this is what we have suitable gene means, right? Now let's write the answer of question number 5. So a suitable gene refers to the specific segment of DNA. A suitable gene refers to the specific segment of DNA which can be injected into the cells of host organism's body. Where this produces the specific protein. Now this protein kills the causative organism of specific disease. It provides immunity to the host organism in which we have introduced the suitable gene. So this is what we have the meaning of suitable gene. This is nothing but a special segment, a specific segment of DNA which is introduced in the cells of the host organism's body, and there it produces a specific type of protein which is helpful in killing the causative agent or causative organism of specific disease. Also it provides the immunity, right? So this is what we have, and with this we have completed the solution of question number 5. Now question number 6 is asking name the primary and secondary lymphoid organs, right? So we have to name some lymphoid organs which are primary and which are secondary in nature. So starting with the primary lymphoid organs, we have two primary lymphoid organs in our body. What are them? So primary lymphoid organs are bone marrow and thymus, right? Primary lymphoid organs are bone marrow and thymus, where the immature lymphocytes differentiates into antigen specific lymphocytes. Now if we talk about what are examples of secondary lymphoid organs, then we have spleen, we have lymph nodes, we have tonsils, Pierce patches and appendix. All these are the examples of secondary lymphoid organs, right? Now let's write answer of this question number 6, starting with the primary lymphoid organs. So primary lymphoid organ are bone marrow and thymus, where the immature lymphocytes differentiates into, where the immature lymphocytes differentiate into antigen specific lymphocytes. Now the secondary lymphoid organs. If we talk about the secondary lymphoid organs, which are secondary in nature, we have some examples such as your Pear's patches, then we have tonsils, appendix, spleen, and lymph nodes. Right? So these all are the examples of secondary lymphoid organs, and bone marrow and thymus are the primary lymphoid organs where the immature lymphocytes gets differentiated into specific antigen specific lymphoids, right? So this is the complete solution of question number 6. Now moving on to the next question. The following are some well-known abbreviations which have been used in this chapter. Now we have to expand each of them to its full form. Alright, so we have given some abbreviated forms and which are very well-known and used in this chapter also. So we have to expand each of them to its expanded form or to its full form. So if we start this answer, then we'll be starting with the very first abbreviated form, that is MALT. If we talk about what is the expanded form of MALT or what is the full form of MALT, then this is mucosal associated lymphoid organ, lymphoid tissue. So this is the expanded form or full form of MALT. Now come with the second form. Let's come with the second form, that is CMI. So the expanded form or full form of CMI is cell mediated immunity. The third term or the third abbreviated form we have to expand is AIDS, AIDS, AIDS. So what is the full form of this AIDS? Acquired immunodeficiency syndrome. So acquired immunodeficiency syndrome. Right? The fourth one, the D one we have is NACO, NACO. For this we have National AIDS Control Organization. National AIDS Control Organization is the expanded form or full form of the abbreviated form such as NACO. Then we have left with the last one, that is E, and HIV is the abbreviated form which we have to expand to its full form. Now if for this we have human immunodeficiency virus. This is the full form of this, human immunodeficiency virus. This is a virus which is actually responsible for causing AIDS in the human population. Now this is the answer, complete solution of question number 7, and we have already completed the full form of each and every abbreviated form we have given in this question. Now in the question number 8 we have to differentiate between the following terms we have given. So innate immunity and acquired immunity are the first one. We have to differentiate between innate and acquired immunity. Alright, so what is innate immunity and what is acquired immunity? We know that innate immunity is the immunity which is inherited from parents, and acquired immunity is not inherited from parents, it is acquired during the lifetime after birth. The most important thing about these two is that innate immunity is the non-specific to pathogen. Innate immunity is non-specific to pathogen, whereas the acquired immunity is specific to pathogen. If you talk about the example, then stomach acid such as HCl and the skin are examples of innate immunity, which is inherited from the parents and is not acquired during lifetime. And if you talk about example of acquired immunity, then antibodies, the antibodies which we take from outside or acquire during the lifetime by entering of pathogen into our body. The production of these kind of antibodies are the example of acquired immunity. So this is the difference between innate and acquired immunity. So let's write the differences between innate and acquired immunity. In the left column we are going to write about innate immunity, and in the right column we are going to talk about acquired immunity, right? Now let's talk about what is the difference between these two. The first point we are taking is it is non-specific. It is non-specific to pathogens. It is non-specific to pathogens, whereas this is specific. This is specific to pathogen, right? Now, this innate immunity is actually inherited from the parents. The immunity which is inherited from the parents is termed as innate immunity, whereas this kind of immunity, that is acquired immunity, is acquired during the lifetime after birth by entering the specific pathogen into body, right? Now if we talk about the example of innate immunity, we have stomach acid, that is HCl. Stomach acid is the example of innate immunity. Then we have skin as an example of innate immunity, whereas the example of acquired immunity is antibodies produced by entering a specific kind of pathogen into your body. This is the example of acquired immunity, and we have completed the difference between innate immunity and acquired immunity. Now let's come towards the next part, that is B part of the same question, that is we have to differentiate between active and passive immunity. So now we are going to talk about the differences between active immunity and passive immunity. What is active immunity and what is passive immunity? First of all, the active immunity is long lasting, is having long lasting effects, whereas passive immunity does not have long lasting effects, right? Now, if we talk about the active and passive immunity, here in the left column we are going to discuss about active immunity, and here we are going to talk about passive immunity, right? In active immunity, it have long lasting effects. It have long lasting effects. Which one? Active immunity have long lasting effects, whereas if we talk about the passive immunity, it does not have does not have the long-lasting effects. Long-lasting effects. Right? Now, the second point to differentiate these two is this immunity, active immunity is produced, the antibodies actually in this immunity, the antibodies against any pathogen produced in the body itself, produced in the body itself, right? And whereas if we talk about in the passive immunity, the antibodies are taken from the outside. The body is not going to make the antibodies. So antibodies in the passive immunity are taken from outside, right? So this is the difference between active immunity and passive immunity. Then if we talk about the example, then antibodies and IgG are the examples for active immunity and passive immunity. So with this we have completed the solution for question number 8. Now moving on to the next question, that is question number 9, and in this question we have to draw a well-labeled diagram of an antibody molecule. Now what is an antibody? Antibodies are a special kind of proteins produced by B lymphocytes in your body in response to antigens. These antibodies are produced or made up of 2 heavy chains and 2 light chains. They are also having interchains and as well as intra-chain disulfide bonds, and the portion of light chain and heavy chain which interacts with the antigen is known as antigen binding site of an antibody. So this is what we have the definition of antibody, or this is what we call what is actually antibody. Now let's make a labeled diagram of antibody molecule on the screen. First of all, we will writing what is antibody. So antibodies are special kind of proteins. These are special kind of proteins produced by B lymphocytes. B lymphocytes in response to antigens, right? These antibodies, these are made up of 2 light chains and 2 heavy chains. Also they are having inter-chain and intra-chain disulfide bonds. Right? Now let's make or draw a well-labeled diagram of an antibody molecule here. So first, if we talk about the diagram, this will be like this. These are first of all Y-shaped molecules. Y-shaped molecules. The second chain is like this. This is your light chain. As we know that antibody molecule is made up of 2 light chains and 2 heavy chains. So this is 1 and this is 2 heavy chains and 2 light chains. This is 1 light chain and the next light chain I am making here. This is your next light chain. Now, we know that in a molecule of antibody there are interchain as well as intra-chain disulfide bonds. So see, here we have disulfide bonds, intra-disulfide bonds like this, and inter-disulfide bonds are also present like this. Intra-disulfide bonds are also present, right? These are disulfide bonds or the representation of disulfide bonds, right? Now, we know that the portion of light chain and heavy chain which interacts with the antigen in the body, this portion is known as antigen binding site. So, let's label this diagram. First of all, we have completed this diagram and this is your antibody molecule. Now, let's label this. First of all, these are the heavy chains. These are heavy chains. This is your light chain. This is also your light chain. Light chain. These are disulfide bonds. Disulfide bonds, and this portion to which the antigen is actually binding, this site is also known, this site is known as antigen binding site. Antigen binding site. Right, so this is the complete solution of question number 9. Now let's talk about the next question, that is question number 10, and this question is asking what are the various routes by which transmission of human immunodeficiency virus takes place. So we have different kind of or different various routes by which actually human immunodeficiency virus goes into the body of an organism and causes AIDS. So what are these routes? First of all, this can enter into the human body by sexual contact with the infected person, right? The sexual contact with the infected person can make the entry of human immunodeficiency virus into the organism. Then the transfusion of infected blood and blood products, this is also one of the reason. Then from placenta it also can transmitted from person to person. That means from infected mother to its child. Last but not least, this also can transmit by sharing needles by drug abusers. So all these are the routes by which actually the human immunodeficiency virus goes into the body of an organism. So these are the routes. So let's write answer of question number 10. Some routes are, first is by sexual contact with infected person. Sexual contact with infected person. The second reason, the second route by which human immunodeficiency virus or HIV can enter into the body of an organism is by transfusion. Of infected blood and blood products. Right, the C is we have by placenta. That means it can transmit from an infected mother to its child, right? The next one, the D1 we have is by sharing needles, by sharing needles of drug abusers, drug abusers. So this is what we have solution for question number 10. Now let's come towards the next question, that is question number 11. What is the mechanism by which the AIDS virus causes deficiency of immune system of the infected person? Alright, if you're talking about AIDS, AIDS is the abbreviated form of a disease which has a full form such as acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome or AIDS is actually caused by a special kind of retrovirus which is known as HIV, or full form for this HIV virus is human immunodeficiency virus. So this is the causative organism, causative agent for acquired immunodeficiency syndrome or AIDS, right? Now what happens actually, or what is actually the mechanism of this virus? So these HIV virus enters into the macrophages of the human body. These HIV virus enters into the host body and they enters into the macrophages of the host organism's body. After entering into the macrophages, the RNA of HIV virus replicates with the help of reverse transcriptase enzyme and produces a viral DNA, right? Now this viral DNA incorporates with the host DNA. At the same time, HIV virus also enters into the T helper T lymphocytes, where it also replicates and forms virus particles. Now these virus particles or this viral progeny actually incorporates or enters into the other healthy helper T lymphocytes. As a result, the T lymphocyte counts in the human body or host organism's body decreases progressively, thereby decreasing the immunity of a host organism. So this is what we have a mechanism of this human immunodeficiency virus, which is actually causative organism for AIDS. Now let's write the answer of number 11. So AIDS, which is also known as acquired immunodeficiency syndrome, is caused by HIV. AIDS is caused by HIV virus. Now this virus enters into the macrophages of host body. The organism in which the human immunodeficiency virus is entering, that person or that individual is actually called as host organism or host individual. So acquired immunodeficiency syndrome or AIDS is caused by HIV virus, which is also known as human immunodeficiency virus. What is actually happening? This virus enters into the human body, the host organism body, and it enters into the macrophages of the host organism. After entering the macrophages, the RNA replicates. The RNA replicates there with the help of reverse transcriptase enzyme. Transcriptase enzyme and produces viral DNA and produces a viral DNA. Now this viral DNA incorporates the host DNA. Right? Which produces virus particles. At the same time, these HIV virus enters into the helper T lymphocytes of the host organism's body. So at the same time, these viruses enters into helper T lymphocytes of host organism, and there also they replicate to form viral progeny. Now this viral progeny enters into healthy T lymphocytes. This results into decreasing number of lymphocytes, thereby decreasing the immunity of host organism. So this is how any human immunodeficiency virus decreases the immunity of a person in which it enters and causes the acquired immunodeficiency syndrome. So this is the complete solution of question number 11. Now let's talk about the next question, that is question number 12, and this question is asking how is a cancerous cell different from a normal cell. Alright, how these two cells are actually differentiated from each other. If we talk about a normal cell, then the normal cell shows the differentiation, whereas the cancerous cell does not show differentiation. The next point if we discuss, which is the difference between normal cell and the cancerous cell, is the normal cell remains confined in the area of formation, whereas the cancerous cells do not remain confined in the area of formation. The normal cells always show contact inhibition property, whereas the cancerous cells do not show any contact inhibition in them. Also, if we talk about the division, then the division in the normal cell is actually in the proper manner or in a controlled manner, whereas the cancerous cells do not show any controlled manner of division, right? So these are some differences between normal cell and cancerous cells. Let's write answer of these two. We have to make a column. In the left column, we are going to discuss normal cells, property of normal cells. And in the right column, we have to discuss the property of cancerous cells. Alright, so if we talk about the first point, the first one we have is, we know that what is normal cells? What is normal cell? If we talk about the first property of normal cell is, they show contact inhibition. They show contact inhibition. That means whenever they come in contact, they stop division. Whereas the cancerous cells do not show contact inhibition property, right? Any normal cell will show you contact inhibition, whereas the cancerous cells do not show any contact inhibition property in them. The next difference between these two is they remain confined in the area of formation, whereas they do not remain confined to the area of formation. The next property of normal cell is they divide in a regulated manner, whereas they divide in an uncontrolled manner, right? The last but not least, the property of normal cell is they show differentiation. And the cancerous cells do not show differentiation, hence they are called as cancerous cells, right? So these are some properties of normal cells and these are some properties of cancerous cells, which is telling you the differences between these two cells. And with this we have completed the solution for question number 12. Now let's come towards the next question, that is question number 13, in which we have to explain what is meant by metastasis. Alright, so we have to give what is actually metastasis. So metastasis is the property or ability of cancerous cells by which the cancer cells actually transmit from one part of the body or one organ of the body to the another organ of the body, right? So these cancer cells, when migrate from one part of the body to the another part of the body, they migrate with the help of blood and lymph pathways, and they invade other healthy tissues of the body and causes or forms new tumors there. This property or this mechanism is actually termed as metastasis, which is the property of only cancer cells. Alright, metastasis is the property or ability of cancer cells in which the cancer cells, with the help of blood and lymph, they transmit from one part or one organ of the body to the another part or another organ of the body. This property is known as metastasis and is shown by malignant tumors only, and is not shown by the another type of tumor, that is benign tumors. The benign tumors are not harmful, but the malignant tumors are very, very harmful because of the property of this metastasis, right? Now let's write the answer on number 13. What is metastasis? So metastasis is the ability of cancer cells to transmit tumors from one part or organ of the body to another organ or part of the body, right? Now these cancer cells actually travels into your body with the help of blood and lymph. So these cancer cells migrate with the help of blood and lymph, pathways, right? So these cancer cells migrate into your body from one place to another place with the help of these blood and lymph pathways, by which they invade the healthy tissues and forms new tumors This property is known as metastasis, right? This property is known as metastasis and is shown by only malignant tumors and not by benign tumors That is why the benign tumors are not harmful, whereas the malignant tumors are very harmful, because the cancer cells of malignant tumors are capable of migration from one place to another place, or we can say that malignant tumor is harmful because of the property of metastasis, and the cells of benign tumors do not have this property of metastasis. That is why they are not harmful, right? So with this we have completed the solution of question number 13. So now let's talk about the next question, that is question number 14, and in this question we have to list some harmful effects caused by drug and alcohol abuse. Alright, so drug or alcohol abuse can cause various kind of harmful effects, not only on individual but on families and in societies also. So how we can discuss it? And if we talk about on individual level, this can affect the nervous system of a person who is addicted towards alcohol or drug, and because of this it can lose judgment and willpower, right? Also, it can show reckless behavior, isolation, depression, weight loss, etc. Also, it can affect dysfunctioning of some organs like kidney, liver, etc. Right? So these are the harmful effects on an individual. Now if we discuss the harmful effects on family, then a person who is addicted towards alcohol, alcohol or drug, can show various issues which leads to frustration and quarrels in the family. And at the level of society, a person who is addicted to alcohol or drug can actually disintegrates or degenerates social network, right? And the person loses interest in the social or any other activities. Also, they can cause some other problems like violence. They can degenerate, they can damage the properties because of their mood swings. So these are the harmful effects. These are the some harmful effects because of drug and alcohol abuse. Now let's write answer of this question, answer number 14. So we will be writing like this, that alcohol and drug abuse, alcohol and drug abuse can cause various harmful effects. Not only on individual, not only on individual, but also on family and in society, right? Let's discuss how. So it can affect, the alcohol and drug abuse can affect the nervous system, of person, because of which he or she loses judgment, willpower, etc. Also, it can show reckless behavior, isolation, depression, weight loss, etc. This also can cause liver cirrhosis, peptic ulcers and gastric carcinoma, right? Also, it can cause dysfunctioning of some organs like kidney, liver, etc. Right? So this is reason on individual level. Now if we talk about the harmful effects on family level, then an addicted person shows various issues which leads to frustration, quarrels, etc. in the family. Now, if we talk about the harmful effects at the level of society, then at society level, an addicted person degenerates the social network, and loses interest in social and other activities activities, right? Also, they can show violence and damage the property, damage the public property due to their mood swings, right? So these are the list, this is the list of some harmful effects because of the alcohol and drug abuse, right? And with this we have completed the solution of question number 14. Let's discuss next question, that is question number 15. In this question we have asked that, do you think that friends can influence one to take alcohol or drugs? If yes, then how may one protect himself or herself from such an influence? Alright, obviously yes, any friend, any of our friend can influence one or us to take alcohol or drugs, right? This is yes, they can influence us. Now, the next is, how may one protect himself or herself from such an influence? So there are some measures, measures which can be helpful for the person to protecting himself or herself from such an influence. What are these measures? These measures are that avoiding undue peer pressure. Avoiding undue peer pressure is one of the measure. Then second is not taking undue pressure of failures beyond its threshold. Also, we can seek medical help, we can take counseling from the counselors, and we can also seek help from our parents and our peers or friends, right? So these are some measures by which anyone can avoid influence of taking alcohol or drugs, right? Now let's write answer number 15. So yes, this is yes. Yes, the friends can influence to take drugs or alcohols. Now there are some measures. There are some measures by which one can protect himself or herself from such an influence. What are these? Let's discuss them. Let's write them. The first one we have is avoiding undue peer pressure. Avoiding undue peer pressure. Then not taking undue pressure from the failures beyond its threshold, right? Then the third measure is seeking help from parents and peers. Then seeking medical help can also avoid such an influence. Then the last but not least, you can take counseling. You can take counseling from counselors, right? So this is the complete solution for question number 15. Now let's talk about the next question, that is question number 16, and this question is asking why is that once a person starts taking alcohol or drugs, it is difficult to get rid of this habit. Discuss it with your teacher. So why this happens when a person starts taking alcohol or drug? It is difficult to get rid of this habit because the consumption of alcohol and drugs has an inherent addictive nature associated with euphoria. What is euphoria? A feeling of happiness, right? So drug and alcohol consumption has an inherent addictive nature associated with euphoria, a feeling of happiness and a temporary feeling of well-being, right? Now, the intake of drug increases the tolerance level of body's receptor, which also leads to the more consumption of drugs or alcohol, right? That is why we find difficulty in getting rid of this habit, right? Now let's write this answer number 16. So the drug and alcohol consumption, has an inherent addictive nature associated with the euphoria and a temporary feeling of well-being. Now, the consumption of alcohol, drug increases the tolerance level of receptors of the body, which leads to the more consumption or intake of drugs. This is why one finds difficulty in getting rid of this habit, right? Now, this is the complete solution of question number 16. Now, let's talk about the last question of this chapter, that is question number 17. In your views, what motivates youngsters to take alcohol and drugs and how this can be avoided? So, generally, youngsters take alcohol and drug out of curiosity, out of excitement. They also take alcohol and drugs because they want to escape from the realities of their life. They want to avoid frustration and disappointments in their life. They also take alcohol and drugs because of their failed relationships, unhappy married lives, right? These are some reasons why youngsters take alcohol and drugs. But it can be avoided by following ways. What are these ways by which we can avoid the youngsters to take alcohol and drugs? We can educate and counsel the child and make them understand, or make them understand to accept the failures, to accept the disappointments as a part of their life. The second is a child should not be pushed. A child should not be pushed unduly to perform beyond his capacity. The third is a teacher and parent should be alert by the activities of child. Also, help can be sought out, a help can be taken from the psychiatrist and psychologist, right? So these are some ways by which we can avoid the youngsters to take alcohol and drugs. With this we have completed, and let's write the answer number 17. So generally alcohol, first of all we will writing like this, alcohol or youngsters, we will write youngsters generally take alcohol and drugs out of curiosity, excitement, to escape from the reality of life, to avoid disappointment, and failures, right? These are some reasons why youngsters generally take alcohol and drugs, but we can avoid by following ways. Let's discuss these ways. The first one is by educating and counseling the child to accept the failures and disappointments as the part of life, Second is a child should not be pushed. A child should not be pushed unduly to perform beyond his capacity. The third one is seeking help from psychologist and psychiatrist. Right, so these are some measures we can take and we can make possible that youngsters avoid taking alcohol and drugs. So with this we have completed the solution of question number 17, and also we have completed this session, this chapter. Right, this is all for today. Thank you and have a nice day.